Imagine an organ in your body that routinely starves its own tissue, triggers a localized explosion just to blow a hole in its own wall, and then, like, aggressively rips up its internal blood vessels just to rebuild them days later.
It sounds incredibly violent.
Right. You'd think we were talking about a massive traumatic injury, but we're actually looking at the baseline, day-to-day reality of human reproduction.
Yeah, and that's what makes today's deep dive so fascinating because, most of our biology is built around maintaining strict homeostasis.
Right, keeping things stable.
Exactly. The lungs, the liver, the heart, their cellular architecture is essentially locked in. But the medical lecture notes and histological diagrams we are analyzing today, they reveal a system operating on a completely different biological paradigm.
A system that's just constantly tearing itself apart.
Right. The female reproductive tract is in this perpetual, relentless state of building, destroying, and remodeling itself, starting straight from the microscopic level up.
And that dynamic architecture, that's our mission for this deep dive. If you are someone who loves to understand how things actually work beneath the surface, you are in the right place. We're looking at the actual cellular mechanics, the histology that makes this entire cycle possible.
And to really understand the scope of this remodeling, the source notes point us straight to the biological source, the ovaries.
Your factory.
Exactly, the factory where the gametes are stored. Now, looking at the physical dimensions, you have these flattened ovoid organs, roughly 3 to 5 centimeters long. But slicing one open under a microscope reveals a very strict division of labor.
Between the inside and the outside.
Right, between the inner core, the medulla, and the outer shell, the cortex. And the spatial organization here tells you immediately what the organ prioritizes.
How so?
Well, the medulla is essentially a logistical hub. It is packed with loose connective tissue and a dense, twisting network of large medullary arteries and veins. It handles the massive systemic blood flow.
Got it. So it's the supply line.
Yeah. But the actual functional units, the ovarian follicles, they are kept strictly in the outer cortex. And there is a notable detail in the clinical texts about the very outer surface covering this cortex.
Oh, right. The part historically called the germinal epithelium?
Yes.
The notes actually flag that name as a complete misnomer, which I found pretty funny.
It really is.
It's just a single layer of cuboidal cells. It's a continuation of the peritoneum. It doesn't germinate anything at all.
No, not at all. The actual cellular action is buried just beneath it in the cortical stroma.
Okay. Let's unpack this, because the population dynamics in that cortex are just baffling to me. I mean, at birth, the cortex holds maybe 500,000 to 1 million primordial follicles, right?
Roughly, yes.
And by puberty, that number drops to around 250,000. But over an entire reproductive lifetime, only about 450 of these follicles will ever successfully mature and ovulate.
That's the staggering math of it.
Right. The hundreds of thousands of others just degenerate through a process called atresia.
Yeah.
And, I mean, biologically speaking, that seems wildly inefficient.
It really does seem that way at first glance.
It's like building a massive state-of-the-art library just to check out a single book a month while systematically burning the rest of the collection. Why do that?
Well, this raises an important question about how biology defines efficiency. In a manufacturing plant, losing 99.9% of your product is a catastrophic failure.
Right, you'd be fired.
Absolutely. But in evolutionary biology, it is a highly rigorous quality control mechanism. The system is intentionally forcing these follicles into an environment of extreme scarcity and competition.
Oh, wow. So they have to fight for resources.
Exactly. Only the follicles that are highly responsive to minute hormonal shifts and possess the most robust cellular machinery survive to maturity. Atresia isn't just passive death, it's a continuous active culling process.
Which perfectly explains the vascular layout we just mentioned. If the cortex was heavily vascularized like the medulla, every follicle would just have easy, direct access to systemic hormones and nutrients.
Right, they'd all get fed.
But by keeping the cortex relatively avascular, the ovary creates a privileged, protected environment. The follicles actually have to actively work to draw in what they need.
They are completely reliant on localized diffusion. It's a tough neighborhood.
Yeah.
And you have to remember, a primordial follicle is essentially dormant. You have a primary oocyte arrested in the first stage of meiotic division, and it's wrapped in a single flat layer of follicular cells.
And it can sit in that metabolically suspended state for, what, decades?
Decades. Literally decades. But when a specific cohort of these follicles finally gets the hormonal signal to wake up, the morphological changes are aggressive.
The texts show the flat cells surrounding the oocyte literally change shape.
Hmm.
They become cuboidal, and then they rapidly multiply into this stratified layer called granulosa cells.
Yep, the growth phase kicks in.
And while that's happening, the oocyte starts secreting glycoproteins to build a thick, specialized wall around itself called the zona pellucida.
The zona pellucida is crucial.
But I was looking at the diagrams of this zona pellucida in the notes, and I was honestly confused because it is a dense physical barrier.
Right.
If the oocyte is completely walling itself off from the granulosa cells, how is it getting the raw materials to grow? I mean, it goes from 25 micrometers to 150 micrometers. It seems like it's building a bunker that will just end up starving it.
Well, the histology here is brilliant because the wall actually isn't solid. As the zona pellucida is being deposited, the granulosa cells extend these tiny finger-like projections.
The microscopic fingers.
Exactly, they're called philopodia, and they extend deep into that glycoprotein matrix. Simultaneously, the oocyte extends its own microvilli outward from the inside.
Oh, so they meet in the middle.
They meet right in the middle of the zona pellucida and lock together using gap junctions.
That is wild. They're essentially running microscopic conduit pipes straight through the brick wall.
Direct physical channels, yeah. This allows amino acids, glucose metabolites, and signaling molecules to bypass the barrier completely.
So it gets fed through the wall.
Right. And that communication is vital because the follicle is undergoing a massive structural reorganization. The stromal cells surrounding the follicle differentiate into a new outer shell called the ecopolliculi.
Oh, that splits into two layers, right?
It does. It splits into a fibrous outer layer, the mica externa, and a highly vascularized inner layer, the theca interna.
The chemical teamwork between these layers is a fascinating piece of the puzzle. The notes say the theca interna cells are the ones with access to the blood supply, right?
Yes.
And they synthesize androgens.
Yes.
But they can't finish the job themselves. They have to pass those androgens across the basement membrane to the granulosa cell.
You're right, because the granulosa cells don't have direct blood access at this stage.
Exactly. So the granulosa cells act as a chemical refinery. They take those androgens and use an enzyme called aromatase to convert them into estradiol.
Which is such a great example of cellular interdependency. Neither cell type holds the complete enzymatic pathway to produce the necessary estrogen on its own.
They completely need each other.
They really do. And as this estrogen production ramps up, the granulosa cells begin secreting a fluid called liquor folliculi.
And that fluid basically just forces the cells apart, right?
Yeah, it creates these fluid spaces that eventually merge into a massive single cavity called an antrum.
And the oocyte just gets pushed to the side. The diagrams show it sitting on a little cellular pedestal, the cumulus oophoris.
That's right.
By the time we reach this final Graafian follicle stage, the structure has swollen to over 2.5 centimeters in diameter.
It's huge.
Keeping in mind the entire ovary is only 3 to 5 centimeters to begin with. This single follicle is now bulging severely off the surface of the organ.
The physical tension on the ovarian capsule at this stage is immense. The tissue is stretched incredibly thin. And this sets the stage for the climax of the whole process, ovulation.
And here's where it gets really interesting. Because I always pictured ovulation as, the follicle just popping like a balloon from high pressure.
That's a common misconception, actually.
But the notes show it isn't just popping. It is a highly coordinated, localized degradation of tissue.
Yeah. The pressure inside the antrum certainly increases, definitely, because the cells secrete proteoglycans and hyaluronic acid, which draw in water osmotically.
Like a sponge.
Right, like a sponge. But the actual rupture is chemical, not mechanical. The blood vessels supplying the very apex of the bulging follicle, a region called the stigma, they constrict.
So the body intentionally induces localized ischemia. It just starves that specific patch of tissue of oxygen.
Exactly. The tissue at the stigma weakens and necrotizes. It dies off. And simultaneously, surrounding cells release proteases.
Those are enzymes that break down proteins, right?
Yeah, they aggressively chew up the collagen network in the ovarian wall, so the structural integrity just completely fails, the wall gives way, and the follicular fluid washes the oocyte, which is still wrapped in its cumulus cells, right out into the peritoneal cavity.
The oocyte is finally out, having just completed its first meiotic division. Viable for what, just 24 hours?
Just about 24 hours, yeah.
But looking back at the ovary, it is essentially just blown a massive hole in its own cortex.
A literal crater.
A literal crater.
Yeah.
And it doesn't slowly heal this crater over weeks. It initiates a radical, immediate recycling program. The ruptured follicle collapses into a bloody clot, which the texts call the corpus hemorrhagicum.
Right.
Macrophages come in to clear the debris, and the remaining granulosa and the Okazeka cells undergo a massive transformation to build an entirely new temporary endocrine gland.
The corpus luteum. And what's fascinating here is the sheer speed and aggressiveness of this structural shift.
Yeah, it's like a stadium hosting the Super Bowl, and immediately after the game, the stadium collapses and instantly reshapes itself into a chemical plant.
That is a perfect analogy. Because for the corpus luteum to pump out the massive quantities of progesterone required to support a potential pregnancy, it needs systemic blood access. Fast.
Which is a problem, because remember, the granulosa cells spent their entire existence isolated behind a basement membrane.
Exactly, they were completely avascular.
They relied entirely on diffusion.
Right. So to fix that, the basement membrane is actively torn down. Capillaries from the Thipulika interna aggressively invade the granulosa
Like
layer.
ripping through the tissue.
Yeah, it's intense. And the cells themselves undergo massive hypertrophy. They swell up to become granulosa lutein cells, which now make up 80% of the corpus luteum mass.
And the theca cells.
The theca cells become the paralutein cells, making up the remaining 20%. And they shift their metabolic machinery almost entirely to progesterone synthesis.
It's a complete repurposing of the biological real estate.
Ooh.
But let's shift focus back to the oocyte. Because the ovary has done its job, the egg is out in the open space of the peritoneal cavity.
Right, it's floating in space.
And we have a massive logistical problem here. Unlike a sperm cell, the oocyte has no tail, no flagellum. It has no internal motor whatsoever. It is a completely passive sphere.
None. It can't move itself.
So how does a passive sphere cross the anatomical gap and navigate a 12-centimeter transit tube to reach the uterus? I mean, 12 centimeters is huge on a microscopic scale.
It's a massive journey, and it relies entirely on the architecture of the fallopian tube, or oviduct. The open end of the tube, the infundibulum, has these finger-like projections called fimbriae.
They kind of look like a sea anemone in the diagrams.
Yeah, they do. And they actively sweep over the surface of the ovary to catch the oocyte. Once it's inside, the oocyte enters the ampulla, which is the widest segment, and usually the site where fertilization actually occurs.
But the histological cross-section of this ampulla reveals it is not a smooth, simple pipe.
Not at all.
The mucosa, the inner lining, is folded into a highly complex branching labyrinth. It looks like a dense forest of longitudinal folds that almost entirely fill the lumen.
It looks chaotic.
Right. If you drop a passive sphere into a dense, fleshy maze, it should just get stuck.
You would think so, but that labyrinthine structure is highly functional. It drastically increases the surface area for the specialized epithelial cells lining the tube.
Okay, so more surface area for what exactly?
Well, you have non-ciliated secretory cells that produce a very specific watery tubal fluid. This fluid is rich in potassium, chloride, and normal serum proteins.
So it's essentially a custom nutrient bath.
Exactly. It creates a highly controlled bath that sustains the oocyte and potentially the sperm and early embryo during the entire transit.
Okay, so that handles the nutritional requirements. But we still need propulsion. How does it move through the maze?
The propulsion is a dual-action mechanical system. Interspersed among those secretory cells are millions of ciliated cells.
Like tiny microscopic brims.
Right. These cilia beat in a coordinated wave-like motion, physically sweeping the fluid and the oocyte toward the uterus.
And what's the second part of the system?
Beneath this mucosa is the muscularis layer. It's an inner circular and outer longitudinal layer of smooth muscle.
Oh, so the smooth muscle is creating peristaltic waves, squeezing the tube, while the microscopic cilia brush the oocyte along the folds.
Yes.
It's basically an active, constantly weaving conveyor belt.
And that conveyor belt is highly regulated by the same hormones we just discussed. Estrogen from the growing follicle earlier in the cycle actually increases the number of cilia and their beat frequency.
Oh, wow.
Yeah, the system is perfectly timed so that the transport mechanics peak right as the oocyte arrives.
Everything is synced up.
Yeah.
Which brings us to the final destination, the uterus. If the fallopian tube is the transit system, the uterus is the canvas for the body's most dramatic remodeling project.
The ultimate staging ground.
Right. The outer wall, the myometrium, is this incredibly thick layer of interlaced smooth muscle. It houses the arcuate arteries within its stratum vasculaire, and it provides the raw mechanical power for labor.
But the inner lining, the endometrium, is where the cyclical histology is really focused.
And the notes explain that the endometrium is strictly divided by function.
Right. You have the deep permanent layer called the stratum basalis. It is supplied by straight arteries and serves as the foundational anchor. It remains relatively unchanged.
It just stays put.
Exactly. But sitting directly on top of it is the stratum functionalis. This superficial layer is supplied by long, highly coiled spiral arteries, and this is the tissue that completely alters its architecture in response to the ovarian cycle.
So, mapping this out, during the proliferative phase, which is roughly days 5 to 14 of the cycle, the growing follicles in the ovary are pumping out estrogen,
Yes, the estrogen levels are rising.
and the stratum bassalis responds by rapidly multiplying. Straight tubular glands grow upwards, and those coiled spiral arteries elongate to vascularize the new tissue. The functionalis builds itself up to 2 or 3 millimeters thick.
And then, following ovulation, the cycle enters the secretory phase, days 15 to 28. The newly formed corpus luteum in the ovary shifts the primary signal to progesterone.
And the uterus just transforms again.
Radically. The endometrial architecture changes dramatically. The simple tubular glands become highly tortuous. They take on a distinct sawtooth appearance under the microscope.
What are they doing at that stage?
They begin actively secreting large amounts of glycogen and glycoproteins into the uterine cavity.
Preparing a food supply. And the tissue itself becomes highly edematous, right? Swelling with fluid and reaching up to 5 millimeters in thickness.
It becomes very thick, very nutrient dense.
So what does this all mean? The metabolic cost of building this complex, highly vascular, nutrient-dense tissue layer is immense.
It's a huge investment of resources.
Why construct this intricate functionalis layer just to undergo menstruation, tearing it down and discarding it, if no embryo implants? Why not just maintain a permanent, thick, nutrient-rich lining ready for a fertilized egg at all times?
If we connect this to the bigger picture, it fundamentally comes down to tissue viability and embryonic signaling.
Okay, unpack that a bit.
Well, an embryo requires an incredibly specific, biochemically pristine environment to successfully implant. A permanent mucosal lining would inevitably age over time.
It would get stale.
Exactly. It would accumulate cellular damage and become much less responsive to embryonic signals.
So the body prefers a completely fresh canvas.
Every single month.
Yes. And the mechanism for clearing that canvas is built directly into the vascular architecture itself. The spiral arteries supplying the stratum functionalis are uniquely sensitive to progesterone.
Okay.
If an embryo does not implant, it cannot send the chemical signal human chorionic gonadotropin that is needed to rescue the corpus luteum in the ovary.
And without that rescue signal from an embryo, the corpus luteum just undergoes autolysis. It self-destructs after about 10 to 14 days.
Right. And when it self-destructs, systemic progesterone levels plummet. The spiral arteries in the uterus instantly detect this sudden drop.
And how do they react? Their smooth muscle walls undergo intense, prolonged spasms. This sudden constriction effectively cuts off blood flow to the entire stratum functionalis. The tissue is literally starved of oxygen and nutrients.
Wow. So it's another localized ischemic event, just like the rupture at the ovarian stigma we talked about earlier.
Exactly the same principle. The ischemic functionalis tissue necrotizes and begins to separate from the permanent bassalis layer.
It dies and detaches.
Yeah. The weakened spiral arteries eventually rupture, flushing the dead tissue away during menstruation.
And what about the base layer?
These straight arteries in the bassalis layer are unaffected by the progesterone drop, so that ensures the foundational layer survives, ready to begin the proliferative phase all over again a few days later.
It's just, the mechanics are relentless. Tracing this from the start, we see a dormant, primordial follicle woken up to build a highly complex cellular infrastructure, relying on gap junctions just to feed the oocyte.
Pushing food through a wall.
Right. We see the dramatic, chemically induced rupture of the ovarian wall, followed instantly by the breakdown of basement membranes to build a highly vascular endocrine gland.
The rapid rebuilding.
We track the oocyte navigating a ciliated muscular labyrinth, ending in a uterus that relies on perfectly timed vascular spasms to violently shed its own lining, all to ensure a biologically pristine environment for the next cycle.
The degree of cellular turnover is just staggering. And, examining the clinical notes leaves us with one final fascinating histological consequence of all this.
Oh, what's that?
Well, when the corpus luteum ultimately degenerates, it leaves behind a dense mass of collagenous scar tissue. It's called a corpus albicans.
A scar in the ovary.
Yeah. The literature notes that over time, macrophages slowly phagocytose and clear the scar tissue. But the process is slow, and they don't always clear it entirely.
So what happens to the ones that are left?
They accumulate. A postmenopausal ovary can actually contain hundreds of these white fiber scars, around 500 of them scattered throughout the stroma.
Wow. It is a physical record of decades of managed trauma.
It really forces you to reconsider how specialized tissues manage long-term damage. I mean, an organ that undergoes a miniature intentional rupture, hemorrhage, and scarring event every single month for 30 to 40 years.
Yeah.
It is operating with a baseline capacity for repair that vastly outstrips static organs. It's a masterclass in controlled biological destruction and regeneration.
A masterclass in controlled destruction hidden entirely from view. I love that. Thank you for joining us on this deep dive into the microscopic world of reproductive histology. It's a compelling reminder that the biology maintaining our existence is rarely static.
It really isn't.
It is constantly tearing itself down to build something stronger. Keep looking beneath the surface, stay intensely curious, and we will catch you on the next deep dive.